All right, so just some announcements we can go through briefly today. So firstly, there's a few new things that are either on Canvas or about to be on Canvas. One of these is a tips document. So this is based on going through 60 mission checkpoints. And these are some things that I saw coming up again and again. So I want to make sure that people are aware of them. And if you follow this advice, then you'll be sure to do better in the semifinals and finals. And then slides guidelines we'll talk about quickly today, and that will also be going on Canvas. So here's some upcoming due dates for the next few days. So you can optionally resubmit your mission checkpoint. This is no obligation, but if you want additional feedback, feel free to do that by Saturday. On Sunday, we have the quiz from last week's speaker that is due, as well as a certificate form. And I really want to emphasize this because we have not gotten too many yet. We've only gotten about 80, and we would expect probably at least double that for people who are seeking certificates. So please fill this out. It's very short, but we need your information in order to issue you a certificate. On Monday, we're asking for a draft of your slides for your pitch. So you have a few days to work on that, and then that'll just be a draft. So kind of something that we can look at and give you feedback. And then next Tuesday, come practice your pitch with us and we can give you feedback as well. So here are some recommended guidelines for your pitch. So we have broken it down into different areas that we would like you to address. The entire pitch will be 1 minute and 50 seconds long. So you need to be efficient and really highlight the most important parts of your mission. So we recommend you have a name for your mission. It's more fun, it's catchy, it'll help people remember it. Everyone on the team needs to be contributing. So we don't want one team member doing all of the work. So make sure everyone's contributing, and we'll be evaluating this at the end of the program to make sure that everyone is eligible for the certificate based on the work that they put in. Don't have too much text. Don't go overtime because we have approximately 60 pitches to do in 2 hours. It's a really tight turnaround. So if you go over time, it's not fair to the other participants. We'll be sharing the slides in order to streamline the process of going from slide to slide. Just say slide when you want us to advance to the next one. We recommend you practice with each other. You can practice with other teams. Just have fun with it so you can really get your pacing down and you won't be blindsided on semifinals day with too long of a pitch. And then also we understand that 1 minute and 50 seconds is really a short amount of time for how much work you guys have put into this. So we're giving you the opportunity to make a fact sheet to accompany your presentation. And we're not going to have a lot of guidelines for this. It's kind of what information you think is important to convey. This is just an example fact sheet I'm giving based on a mission concept study that I did last year. Some of the important information is conveyed, but again, it's up to you what you want to put on it. And it doesn't have to be, there's no template. It's just whatever you think is important can go on your fact sheet. And with that, I'm really excited to introduce our speaker, Dr. Sudha Rajamani, who is speaking to us from India. So big thank you to her because it's quite late at night over there. And so just a bit of background on Dr. Rajamani. So she actually didn't begin astrobiology until she was a postdoc at UCSC, which is in California. And then her interest in the field and especially origins of life really began to grow. She then worked at Harvard and then returned to India to join the Indian Institute of Science, Education, and Research. So she's now an associate professor there and the principal investigator of the Chemical Origins of Life Lab. So they use a variety of tools and disciplines there to delineate the processes and niches that would have allowed for the transition from non-life to life on early Earth. She's also very passionate about astrobiology, education, and outreach, and she's involved in this formally and via science outreach programs. And she does a lot of mentoring of school and university students in pursuit of their astrobiology careers. And so we're very excited to have Dr. Rajamani with us because her goals are very aligned with our goals as a foundation. And we're excited to hear about her work in this field. So thank you, Dr. Rajamani.
Thank you so much, Tess. And thank you all at Womanium for having me. As Tess mentioned, I'm beaming in from Pune, and I'm based at this institute called the Indian Institute of Science, Education, and Research, where for about 11 and a half years, my team and I have been trying to basically undertake experiments to answer one of the most profound questions that's out there, which is to try and understand how life came about on Earth. And this story of life's origin and its early evolution, basically what I'm going to talk to you about today is to give you the context of how we think about questions in this area and how astrobiology as a science allows us to understand the origins, the evolution, the distribution, and potentially the future of life in the universe. So without further ado, let's get going. None of this would have been possible without a whole bunch of people who really love working with me and working on these interesting questions, help out with all of these things. And many of them are graduate students, some of them are integrated PhD students, but there's a whole bunch of undergrads who go through the lab. And we call ourselves the cool lab because we essentially are trying to understand how chemistry might have transitioned to biology on the early Earth. So when I talk about this, when I talk about trying to understand the origins of life, one needs to start out with a definition that you can work with. So one of the working definitions that was ratified by a committee that was put together by the NASA Astrobiology Institute way back in '92 basically defined life as a self-sustaining chemical system capable of evolution. And when we talk about the hallmarks of life, some of the most fundamental hallmarks that are common to all life as we understand and know it includes compartmentalization, where you have this boundary that holds a whole bunch of molecules and chemicals together, where very interesting and fun chemistry and biology happens. Life basically requires a means of propagating itself. So self-replication is a very, very important feature. And you need to have a metabolic process in place, set of metabolic processes in place that allow life to exist far from equilibrium, which is a very, very important hallmark of life. So when we think about these basic features of life, one then starts to wonder, how can you start thinking about this transition that I'm talking about, which we think might have happened about 4 billion years ago, this transition from non-life to life, or from chemistry to biology. And when I say that, where am I coming from? I'm coming from the fact that there is this absolutely beautiful continuum that exists between physics, chemistry, and biology, as has been sort of outlined in the slide. So if you look at the bottom panel, let me just try and get my pointer here. Yeah, if you look at this bottom panel, this is where you're seeing molecules that exist in the biological realm. Some are small and they're called monomers. They come together to form much larger molecules called polymers. And in this particular case, I've used two examples to illustrate this. One is cytochrome C, which is a protein, a fundamentally important protein, which goes back to some of the oldest forms of life that plays a central role in electron transport. And on the right side is another molecule, which is made up of ribose nucleic acid, which is a cousin of deoxyribose nucleic acid, which essentially encodes information in our cells. And both these molecules do something amazing. They facilitate these grand reactions which basically catalyze a lot of interesting processes in the cells. If you zoom into them, you basically are getting into this realm of the monomeric molecules in the biological space. Like here I'm using 4 examples, all 4 of them are that of amino acids, but it could be any number of fundamental monomeric units which make up other classes of biomolecules, including the nucleic acids, carbohydrates, and lipids. But zoom into one of these molecules, and what do you see? They are made up of smaller chemical entities. And this kind of, these chemical entities are very readily plausible even in the interstellar medium, where you have extremely interesting chemistry going on, which produces some of the fundamental molecules. You zoom further into them, what do you see? They're made up of atoms. Atoms come together to form molecules in the chemical space. The smaller molecules in the chemical space come together to form the monomeric entities in the biological space, which then come together to form polymers, which suddenly now can do things that you cannot ascribe back to the individual monomers. This kind of a property is called emergent property, and we think this is fundamentally crucial for the transition to have happened from non-life to life. So this continuum allows us to now think about this problem of this transition from chemistry to biology in a very systematic fashion. So where do we think these biomolecules on the Earth came about from? There are 2 ways to think about this. One is exogenous delivery. It happens even today on our Earth, but back then, very early on in Earth's history, there seems to have been a period when there was extremely heavy bombardment of comets and meteorites, and a lot of these interesting chemical molecules that form in the interstellar medium can ride in these entities and get delivered onto Earth. Another very readily tangible way of thinking about this is to think about niches on the early Earth, which would have been rife with chemical and thermal fluxes, where you could have really interesting synthesis happening given smaller ingredients, where you could now form the biomolecules that I've kind of just listed here. This is really remotely not exhaustive. This is just to give you a flavor of the different kind of molecules that we think would have set the stage for what we call as the prebiotic chemistry phase of emergence of life. So given that in a niche like this terrestrial volcanic geothermal niche that I'm showing here on the right side panel, let's say you had all these really relevant biomolecules present that were fundamentally crucial for this transition to occur, how then can you think about going from this space to life as we see today? So here is a slide where I'm showing you a whole bunch of different living entities, all the way from Nelson Mandela to single-cell bacteria, to a giant catfish, to giant sequoias, to mushrooms, to T-Rex. What is it that binds all of these living entities together? What is sort of the least common denominator that exists in all of them? I'm not sure I can hear you respond right away, so I'm going to give away the answer. The least common denominator that binds all of them together is cells. So one of the interesting questions then is to ask, how do you go from the prebiotic soup of molecules to the oldest cells that might have populated Earth much before even your single cellular prokaryotic entities like bacteria came about? So here is a slide to just give you a sense of how complicated even the most simplest form of unicellular life is. On the left, you have a prokaryotic cell, and one of the most famous examples is that of bacteria. On the right, you have eukaryotic cells, which essentially are the kind of cells that your body and my bodies are made of, and not just us, even fungi, animals, and plants. I think it's readily appreciable that even a unicellular entity is super complex, and the images here are a sort of a minimalized version, cartoon version, to give you a sense of how complicated it might be. So despite them being single-celled in nature, bacteria, for example, can do something amazing, right? They basically can populate every extreme niche that you can think of on modern Earth. And here are examples where you can see that, you see all these colored, orange-colored features in the rocks here. These are actually microorganisms. And this particular rock is exposed to extremely high UV, but these organisms still survive. You and I, we cannot survive in those conditions for a very long time at all. Then you have extremophiles called thermophiles, which are heat-loving organisms, and they can exist in temperatures which are 70 degrees, 80 degrees, 90 degrees centigrade. Then you have salt-loving organisms, which can tolerate very high amounts of salt, where you and I will shrivel up and completely vanish. And you have permafrost-dwelling organisms, where they can live at really, really cold temperatures. So what I'm trying to say is, despite being single-celled in nature, it can occupy every niche, extreme niche that you can think of on modern Earth. So then, given what we understand about some of these extremophiles and what we appreciate from the geological history of Earth, from the cosmology of that kind of set the stage for the planetary systems to come together, and from the prebiotic chemistry that we think might have occurred on the early Earth, you can use both a bottom-up and a top-down approach to converge on understanding how we think the transition from non-life to life might have occurred on the early Earth. So what might have this earliest life forms, what might have they looked like? So this is an artistic rendition, the protocells that I'm showing you here. Essentially, these are, sorry about the spelling error here. If you take a tennis ball and you sort of make this, you kind of cut open a slice, you're looking at the inside of the tennis ball. It's the tennis ball's outer membrane is made up of lipids, and lipids are essentially biomolecules that are the membrane coverings that line your cells and plant cells and bacteria, and name a life form, and it basically is the membrane boundary. And inside it, maybe there was a minimal metabolic network that allowed it to sustain itself, and there was a genetic molecule that encoded information that basically allowed it to propagate itself. And it's thought that the protocells, essentially, there was a stage where a common community of ancestral cells paved the way to how what we see today in life, which is the base of the tree of life that is shown here. In this particular case, I'm using an example of cyanobacteria. So if you look at when we think these prokaryotes originated, if you take the early history of life on Earth and put it on a 24-hour clock, we think that they originated about 3.8 to 4 billion years ago. Some of the oldest geological evidence that exists today is what is shown in this panel here. These pillar-like structures are called stromatolites. These are essentially formed as a consequence of interaction between life and geology. And if you slice one of them open, you see these alternate striations of gold and sort of black regions, which basically is a result of the crosstalk between cyanobacteria and the rock that they were sort of embedded in. And these are some of the oldest evidence, geological evidence that exists today that can be traced back to about 3.5 billion years ago. It's thought that roughly by noon on this 24-hour clock is when these prokaryotes emerged. And when they emerged, what did they do? They did something super important that set the stage for complexification of life. They basically oxygenated all of Earth's atmosphere, which was extremely anoxic to begin with. It was predominantly composed of nitrogen and carbon dioxide before these organisms came about, which then used a process of photosynthesis to oxygenate all of Earth's atmosphere, which eventually, about 2 billion years ago, led to the emergence of cells that make up our bodies, which are the eukaryotes, which is about close to late afternoon on this 24-hour clock. Where did we come about? Just a little before midnight. But what I'm going to focus on today is what we think might have happened about 4 billion years ago, and how do we systematically think about some of the stages that led to the formation of some of these most primordial of cells called protocells. So this is sort of the cartoon representation of tree of life as most of our textbooks might denote even today. The 3 domains of life are shown here: bacteria, eukaryotes, and archaea. At the base of the tree, you have what's called LUCA, or the last universal common ancestor. And before that, we do not know what might have been. This is also a hypothetical entity. We still do not have a systematic proof of LUCA. Over the last couple of decades, the tree of life has undergone dramatic changes because of all the appreciation that has happened from various areas of biology. And what we now think might be more realistic is that at the base of these 3 domains, at the base of the tree, there probably was a common ancestral community of primordial cells that interacted with each other and were lifelike, and basically the oldest forms of living entities that would have probably come together to essentially then lead to all of life that we see today. What can we do to understand how these entities came about? We can systematically look at evidence that is generated from various areas of sciences, including chemistry, including the geological history of early Earth. And like I said, what the oldest forms of life, like the extremophiles that I showed you, what do they reveal to us about what is their fundamental makeup? How do they survive these extreme conditions? So by collecting all of these diverse evidence, I'm happy to say that after close to a century of a lot of effort from a lot of colleagues in the field, the story is finally coming into view, and we are beginning to understand how the pieces of this puzzle sort of can be placed together. Of course, the whole puzzle hasn't come into view yet, but we have understood a lot more about how this process might have happened than we understood, say, 70 years ago. So astrobiology as an area of science, because I think it's a very diverse audience today, I would like to say, is an extremely interdisciplinary area of science that feeds off of inputs from various other areas of sciences. And it's not sort of an exhaustive list that I've mentioned here, but predominantly you have fundamental inputs from astronomy, geology, planetary sciences, physics, paleontology, which tells us about some of the oldest forms of lives, biology, which essentially talks to us about what living systems are all about, and all of the various inputs that we get from colleagues from these various areas of sciences feeds into this fundamental understanding of how we think life might have come about 4 billion years ago. And in my own group, we use a whole bunch of tools from various areas of biological and chemical sciences, including biochemistry, molecular biology, some physical tools rooted in biophysics, but also we take help from colleagues who do fun experiments on the computer, and they basically model many of these things for us because the complexity of the space is very vast. And we kind of use these collective tools to fundamentally understand the question that is sort of lays right here. How do you go from the prebiotic soup to the first cells? So the hope is that, we'll be able to answer this and in our lifetimes. But yeah, let's talk about the early history of life on Earth, and sort of the stage, the period around which this grand drama played out. And we think about 4.2 billion years ago, a stable hydrosphere might have been possible on the early Earth. Very quickly after that, a lot of interesting prebiotic chemistry might have allowed for the formation of the fundamental building blocks of life molecules. How we go from this prebiotic space to a space which is more, which is rife with molecules that we recognize, is something that we are still trying to understand. And in the last, I will say 20 years, there's a better understanding that going directly from this prebiotic chemistry space to what we call an RNA world space, where it is thought that the earliest forms of life used one molecule which could do two functions, which is super cool. It not just encodes information, for example, like the SARS-CoV-2 virus, which is a RNA virus. The RNA molecules are also capable of facilitating extremely cool reactions because they can, unlike their cousin DNA, fold back on themselves and form these very interesting pockets where other molecules can bind and fun reactions can happen. So it's thought that if you can simplify the problem of understanding this transition, you can actually ask this question of how did you go from a prebiotic chemical space to an RNA world? And work in the field that has happened in the last couple of decades has made it very clear that this transition would have been non-trivial, and maybe the RNA world was predated by a world where the molecules that came about, informational molecules and the catalytic molecules that came about, were possibly chemically more simpler, which eventually led to the evolution of molecules that we see in modern biochemistry. So where do we think all of these grand reactions happened? Depending upon whom you talk to in the field, there are two major hypotheses. One thing common to both these hypotheses are it involves volcanoes. One exists on the ocean floor, the other exists on terra firma. So the one on the left panel here is essentially a hydrothermal vent. In this particular case, this is an example of a black smoker because of the kind of interesting chemistry that happens between the water and the rock system there. The resulting fumes that it exudes is black in color. A lot of interesting iron-based chemistry happening there. On the right-hand panel is a terrestrial geothermal pool. And this is actually, I don't know how many of you have been to Mount Larsen National Park in California. This is Bumpus Hell. And Bumpus Hell is a very interesting example of a terrestrial geothermal pool where you have wonderful chemical and thermal fluxes that potentially allow for these uphill reactions that occur when you transition from chemistry to biology. And what do I mean by that? These are just examples of two other similar terrestrial volcanic geothermal pools. What do I mean by that? How do you go from chemical molecules to biological molecules? And how do you even start thinking about this when you have to think about life originating on the early Earth? All good things start with a wonderful theory. And the most famous theory in this context came from two gentlemen who back then did not have internet or no ways of talking to each other. They independently came up with the same hypothesis theory. It's called the Oparin-Haldane Theory. Oparin was in Russia, Haldane was in the US. And basically what they posited is that given the right set of conditions and the right ingredients, they presumed that early Earth was very reducing in nature, a lot of interesting molecules to give away a lot of electrons. So if you had molecules like ammonia, carbon dioxide, water, if you had all of these molecules in a primordial soup of the early Earth, given the right energy conditions like lightning in the atmosphere, ultraviolet radiation from the sun, all of these would have provided very interesting energy sources. You could, in principle, go from some of these chemical entities to what now start looking like biomolecular monomers. In this particular case, they went on to hypothesize that you can actually form simple hydroxy acids and other kinds of simple molecules that then could have come together to form much more complex molecules. And Haldane is attributed basically attributed with coining the term primordial soup. And a theory is a theory till somebody systematically can prove it, systematically test it and see if it holds water. And that particular important experiment was done by Miller-Urey. It happened in the same year when DNA double helix was described. The structure of DNA was described. This is 1953, which is when this is Stanley Miller. He joined Harold Urey and convinced Urey that he wanted to try this experiment. He put together this setup here, right here, that you see Stanley Miller sitting with. And he did what is called the very famous spark discharge experiment. here is the primordial soup that he was trying to simulate. So basically the primordial ocean with a lot of water. And you had these fundamental ingredients, essentially molecules that would be present in a very reducing environment, methane, ammonia, water, dihydrogen. And early Earth was not remotely a beautiful, pleasant place like Earth is today. Despite the extreme environments that we see now, we still know that Earth is a very, very temperature-wise conducive place for life. But early Earth is thought to have been really hot. Depending on whom you talk to, it would have been as warm as 60 degrees to as hot as 100 degrees or a little over that. So basically, they simulated a hot early Earth, primordial soup, containing the ingredients. And when you reach 100 degrees, water vaporizes, carries these molecules, and it takes to this place where you have a tungsten electrode that basically provides this high energy, crazy amount of energy that we think would have been provided by lightning or UV radiation on the early Earth. And that particular interesting high energy atmosphere allows for very fascinating chemistry to happen in the space. And then you can have those vapors condense into liquid again, because you have a condenser here that is seeing cold water being circulated, and the vapor condenses into liquid, and you can essentially collect it from this stopper here, and you can basically get this reaction to keep going over and over again. And what Miller very systematically did is carry on this experiment for many numbers of cycles and systematically collecting samples after various time points. And he used some very, very, very amazing but simple techniques like thin layer chromatography, which is a very, very simple way of separating molecules based on various properties of the molecules, to show that you could now actually have molecules that are seen in biology. And here's glycine, glutamic acid, alanine, serine. These are all amino acids that make up your proteins. And then you have even more simpler molecules like hydroxy acids and urea, which initially was thought was inorganic, but then once it was found that your urine is made up of urea, you knew that biologically urea could be produced. So this particular experiment finally provided a proof that the Oparin-Haldane theory was indeed plausible, and you could systematically think about how you can go from inorganic molecules to organic molecules. This set the stage for a field of research in astrobiology called prebiotic chemistry. And since then, a lot has been understood about different biomolecules that are seen in biochemistry now. And in order to understand how you go from monomeric space of all these interesting monomers of amino acids, monomers of proteins, monomers of nucleic acids, monomers of carbohydrates, to actual cellular entities, you need to work with some model that you can systematically test. One of the famous models that's out there for the terrestrial origins of life is what is described in the slide, and I'll walk you through it. So the idea is that if you had the fundamental ingredients of the biomolecules, for example, these are called fatty acid. This is fatty acid number 1 molecule. Number 2 is a fatty alcohol molecule. These are essentially single chain amphiphiles, which are the fundamental units of the lipids that basically form your membranes. So it's thought that back on early Earth, we would have had just single chain amphiphiles, not the kind of more complex lipids that we see in our cells today. If you have them, and if you had the monomers of the informational or the catalytic molecule present in a tidal pool like this, where very interesting wet-dry cycles driven by seasonal changes, diurnal changes of day-night cycles, basically also rain evaporating down, and then early morning, in the morning when the temperatures are high, water evaporates off and then condenses. These kinds of very interesting wet-dry cycles, which happen even on modern Earth, would have been readily facilitated on the early Earth, which allows for something fascinating to happen. These molecules, which have kind of double face, for example, this hydrocarbon chain hates water, but the head loves water. It's a polar group. So what happens is, the second you take a bunch of these molecules and put them in water, the water-hating groups come together and the water-loving groups stay outside. And this results in the formation of a lot of interesting higher-order structures called micelles and bilayers. And bilayers, when they become long, they flop back and they form these very nice compartments called vesicles. And these vesicles are very reminiscent of the protocells that I was talking to you about. So you would have very readily had these higher order structures form, and all these red little bacteria-like things are essentially monomers of, for example, RNA molecule. So every dehydration phase, you would have had water go away, and you would have had these monomers now trapped between the collapsed layers of the lipid. So now you go from this three-dimensional space to a two-dimensional space. You're constraining the molecules, bringing them closer together, increasing the effective concentration, pulling out water, which does something super important, which essentially gets the monomers to join together to form a polymer. Polymers that exist in our cells predominantly are the outcome of an important chemical reaction called condensation. Condensation reaction is where two monomers come together, water molecule is pulled out, and you essentially have a bond formed. In the case of peptides or proteins, the bond is called, in the case of peptides or proteins, the bond is called a peptide bond. In the case of nucleic acids, the bond is called a phosphodiester bond, and so on. So every dehydration phase, you can essentially, in principle, go from a monomeric space to a polymeric space. And a subsequent rehydration cycle, once again, brings in a lot of water. And now you have these lipid membranes, which have these water-hating groups, immediately bleb out into vesicular compartments, these protocell-like entities, which now encapsulate with them the polymers within the interior, because the interior has water in it. So that is a water-loving area. So the polymers, which are polar in nature, are now encapsulated. And it's thought that some primitive version of selection and multiple rounds of these dehydration and rehydration would have led to the formation of the primordial cells or protocells that we are talking about. So given this model, what are the steps we can think about going from the prebiotic soup to the protocell? The very interesting prebiotic chemistry that I talked to you about, which we saw in the Miller-Urey experiment, would have led to the formation of interesting building blocks. And the model that I described to you now, where these wet-dry cycles would have been readily facilitated, would have essentially allowed for the oligomerization of these monomers to result in either an informational polymer like nucleic acid or catalytic polymers like small peptides and maybe small proteins, not really proteins, but many small peptides. And once an interesting molecule came about in the space which could do something really cool, it's extremely important to invoke or think about ways in which they could have propagated the information encoded in them so that they can survive and continue to be around to do their function. And along the way, you would have had minimal enzymes form from small kinds of entities, starting all the way from metal ions, which then got complexed with other interesting scaffolds. I'll talk about that in a slide to give you a better flavor of that. But they would have brought in the early metabolic flavor to this whole pathway. And I already talked to you about primordial membrane molecules. We still do not know what complexity was entailed in the prebiotic soup. It's suffice to say, it would have been a very heterogeneous mix of molecules. Why biology ended up using the molecules that we see today, while a whole bunch of them would have been possible, is not very clear, but it is reasonable to assume that a lot of selection pressures over these billions of years, especially in the early billions of years when chemical evolution was happening, would have shaped the prebiotic molecular space to eventually lead to a bunch of molecules that emerged victorious and propagated further to form the primordial cells. So for some of the people that are slightly in advanced science courses, one way to think about how do you go to RNA molecules, functional RNA ribozyme molecules, is to try and understand how the prebiotic pool of nucleotides came about. And I'm very happy to share that a whole bunch of colleagues in the field of prebiotic chemistry have described very, very relevant and plausible pathways that would have allowed for the formation of these molecules. We also know how these molecules could come together and form polymers of various chain lengths. Once they go to a certain length, they become much more interesting because they can start folding back on themselves and doing fun stuff. Please appreciate the fact that all of this happened in the absence of enzymes that otherwise do everything in modern biochemistry. And this is a very important thing to consider, because the second you invoke the presence of enzymes, you're now dealing with a chicken and egg problem. You need information encoded in a nucleic acid to make a protein, which can act as an enzyme. But you need a whole bunch of enzymes to make the nucleic acid in the first place. So what came first? It becomes very complicated. It becomes a chicken and egg problem. So if you just relegate proteins to the side and think about, I mean, don't forget they're super, super important, but we don't have to think about complex proteinaceous enzymes. We can just think about simpler, smaller oligomers and how they might have readily come about just driven by chemistry and geology. You can think about, you can describe processes by which you can form polymers of different lengths and sequences. And we can also describe ways in which we think information transfer might have been possible completely in the absence of enzymes, leading to a whole diverse pool of interesting molecules from which we think some of the molecules evolved interesting functions or just randomly had interesting functions come about, which then complexified the possible reactions that might have been feasible under those conditions. So this is one way to think about origins and replication of polymers. I'm using RNA world here as an example, but it's not the end all of the worlds out there, because this completely disregards the contribution from amino acids and peptides, and I'll talk about that later. So we sort of think about the step of going from prebiotic soup to an RNA world in a systematic manner, and this was described by a colleague in the field, Nick Hudd, where they beautifully sort of talked about how your modern nucleic acids, RNA, DNA, would have been predated by what might have been pre-RNA and proto-RNA. And what is this proto-RNA? Think about this for a second. What's your nucleic acid made of? It's made up of nucleobases, which essentially are the main reason information propagation is possible, because they can hydrogen bond with their partners. A base pairs with U, G base pairs with C, and vice versa. So you can basically call them the recognition unit, or RU. You have the ribose molecule here, which is essentially a molecule that connects three different entities. On the one hand, you have nucleobase. On the other hand, you have the phosphate linker, which is the backbone. On the third hand, you have a phosphodiester linkage with which it's connected to the neighboring monomer. So you can call this the trifunctional connector. And the third entity that is important for the molecule to be informational is an ionizable linker. In nucleic acids, the ionizable linker is the backbone, which is a phosphate molecule, which has a net negative charge. So if we now have to think about a proto-RNA, which does not have any of these entities that we see in RNA and DNA today, all you need to think about is molecules that could have played the role of a recognition unit, molecules that could have played the role of trifunctional connectors, molecules that could have been the ionizable linkers that essentially allow these molecules to interact with other nucleic acids or proteins. So this is one way to think about, it's a hypothetical evolutionary lineage. Have we been able to describe some of the steps? The answer is yes. Have we figured all of this out remotely? The answer is no. But a whole bunch of people in our lab actually are very interested in understanding how these pathways might have come about and what these primitive or primordial genetic polymers might have looked like. Sorry, primitive has a spelling error there. And how do we then think about the compartment? And this is just to give you again a flavor. When you have to think about the emergence of protocellular membranes, you need to be able to think about all possible amphiphilic molecules that might have populated the prebiotic soup. Some of the most studied examples involve fatty acids, but there's no reason to think that fatty acids were the only molecules that amphiphilic molecules that might have been present in the prebiotic soup. So what other kinds of interesting chemical amphiphiles would have been present is something that we think about both experimentally, and we also use computational means to understand the space. And given the right conditions, the pH is extremely important because that will determine the nature of the higher order structure they form. And by a process of self-assembly, the second you take this bunch and put the bunch of molecules and put them in water, they literally self-assemble into one of the three higher order structures I talked to you about. One of the most important higher order structure that's relevant to understanding the emergence of protocells is a vesicle. And we think about how a complex but robust vesicle could have come about, which would have tolerated a lot of these harsh physicochemical parameters that might have existed on the early Earth, pH-wise, temperature-wise, the various divalent cations. Why are divalent cations interesting? I can talk about that in the Q&A session. But we think about selection pressures that could have shaped this landscape, which eventually allowed for the formation of the earliest robust protocellular membranes, which could have encapsulated a genetic material and some minimal metabolic network. Once they came about, it's important to think about how did they evolve into what we see today, or at least close to what we see today. And we think about this in a very systematic manner because modern cells, if you look at them, the cell membrane is just not made up of lipids. It's got proteins, it's got other kinds of molecules, for example, cholesterol molecules, that is extremely important for the membrane to behave the way it behaves. And this compartment does extremely fundamental roles in keeping the molecules that are present on the inside, helps them interact, generate, undergo important chemical reactions that produce waste. The waste has to be shuttled out. The shuttling of molecules across the membrane happens via pores that are formed by proteins. So you see, modern membrane is really complex. So one interesting way to think about early functional membranes is to try and think about a pathway where you can go from primordial simple compartments to more complex compartments, which now would have incorporated short peptides in their membrane, or RNA sequences in their membrane, or scaffolds in the membrane like cholesterol, which would have allowed for very interesting functional possibilities. One last thing I kind of will leave you with, which I told I'll talk about in a second, is what might have the oldest catalysts been like that facilitated reactions. Look at your modern, highly engineered molecules called enzymes. They are just made of these very, very long stretches of amino acids that are joined together into this long polypeptide chain that fold back on themselves, are stabilized by secondary and tertiary structures. But this is just too complex to come about from the prebiotic soup. So what do we think might have been much more readily possible? So if you slowly work your way back, the core of a lot of these enzymes basically have metal ions, which either catalyze reactions or are fundamentally crucial to stabilize the structure that catalyzes reactions. So it's thought that maybe hydrated metal ions by themselves could have catalyzed some of the simplest of reactions back then, albeit in a shabby manner, but they would have nevertheless been able to do it. They could have then, for example, if you take your hemoglobin molecule, which essentially binds oxygen at this area and then releases it to the cells, has this porphyrin ring. And porphyrin is one of the important examples of a scaffold of proteins which coordinates with metal ions to facilitate the most fundamental of all reactions there is, which is to bind molecules and release molecules. So maybe some of these scaffolds complexed with metal ions to form these metal complexes, which could now catalyze reactions even better than just hydrated metal ions. And once they started interacting with small peptides that could have readily formed under early Earth conditions, maybe you would have had minimal enzymes come about that look a lot like your modern enzymes, but are much, much more simpler, which could have done some of these functions even better. So this is, again, sort of a hypothetical pathway, and there is reason to believe that you can systematically sort of try and understand how do you go from hydrated metal ions to really complex enzymes. So this is to give you a flavor of how we can think about some of these steps. Happy to state that a recipe for life itself, is it possible to have existed? The answer is yes. And this is coming from several decades of systematic research, both experimental and computational, that has allowed us to appreciate how do you go from fundamental inorganic ingredients to amino acid precursors that essentially led to the formation of proteins, nucleotide precursors that led to the formation of nucleic acids, and amphiphilic precursors or lipid precursors that led to the formation of membranes. So we are at a stage where we understand a good number of these processes reasonably well. But how do you go from the prebiotic soup space and from these biomolecules to what is a cell-like entity is something that's been a bit more challenging, but a lot more has been understood in the last 15 to 20 years than we've ever known before. And all of this knowledge that we're gleaning from trying to understand how life might have come about on the early Earth has fundamental implications for answering this question. Is there life in the universe? Is there any life out there? We do not know. It's still unclear. But there's a whole bunch of exoplanets, which are these other worlds orbiting stars other than our sun, which are of various different kinds. They can be like Jupiters, they can be like Neptunes, they can be more like Earths or super Earths. And there's reason to believe that given the atmosphere, right atmosphere, and the presence of water and some of the interesting chemical entities that I talked to you about, maybe, just maybe, you can understand if there is presence of life in the universe. I think these are things that have been talked about by certain other speakers, so I'm not going to talk about it. But one very important thing is this habitable zone concept, or the Goldilocks concept, which talks about the planet being just at the right distance from its star, where it's neither too hot nor too cold. And given that it has a suitable atmosphere, it could hold water. And that means the possibility of it harboring life is something that we can consider. But the signs of habitability is not equal to signs of life. Extremely important to remember that. Biosignatures are something that allow us to systematically understand this. We study a lot about this on Earth, and we try and understand if you can actually look for these substances, patterns, processes that could indicate the presence of life in extraterrestrial environments, both within our solar system and outside. And what is very important to remember is life, basically, presence of life leads to global modifications. And that's shown in this illustration here. This is, of course, a real picture of the Earth. And this is an artistic illustration of what Earth might have looked like 3.8 to 4.1 billion years ago. And you can very quickly appreciate that the emergence and evolution of life has completely changed the way our planet looks today. So presence of life changes the planet's atmosphere, its surface, basically its fundamental appearance over time. And so looking for biosignatures in exoplanets is very readily possible. And they need to be interpreted in the context of the planetary environment of that particular entity. The geological history of that particular exoplanet could have been very different from Earth's history. And all of this allows us to do this, which is what Curiosity is doing now. Perseverance is doing now. I'm sorry, Curiosity did it a while ago. This is Perseverance rover, fitted with all kinds of gadgets that will allow you to study both the climate and the geology of Mars, to essentially finally help us understand whether Mars in its distant past might have been able to harbor life. The way to think about how these various gadgets can look at signatures is to basically try and understand how life might have emerged on our planet, whose history we know the best, and take all of that knowledge and feed that to all of these various gadgets to systematically understand whether you can look for signatures, atomic molecular signatures on these extraterrestrial environments that will allow us to appreciate whether a particular entity could have harbored life. Thank you for your time, and I will leave you with that slide.
Thank you so much. That was excellent. I really love the way you tied it all together at the end. So if anyone has questions, you can feel free to put them in a Discord. There's already been a few that we can start with. So Subsankar is asking, can the origin of life on Earth be a combination of both the exogenous delivery and endogenous synthesis?
Oh, absolutely. And I just think I want to clarify one thing here. When I say exogenous delivery, my big focus was on the molecules that can ride on comets, meteorites, that constantly would have been bombarding early Earth and early Moon, and would have brought in a lot of this very interesting chemistry that was already happening in the interstellar medium. Yes, that was super important. Some of the organics that came, that rode in these molecules, also had other important ingredients that would have undergone very interesting chemistry in certain niches on the early Earth where they would have been feasible. So you need to think about these niches a little bit systematically, because you need to be able to go from simple molecules to more complex molecules. And most of the times going from simple to complex molecules means making bonds, which means knocking things out of place and bringing things together and stuff. So any niche that can facilitate some of these very, very interesting uphill reactions would be kosher. So I think both would have been very important for populating the early Earth with very interesting molecules relevant to biochemistry.
Great answer. Thank you. So Aditya is asking, did the late heavy bombardment event wipe out all possible life on Earth prior to 3.8 GA? Is it possible to correlate life before and after the event, considering it also evaporated all hydrosphere on Earth during the time?
Yeah, that's a very, very important and relevant question. Like I said, the oldest geological evidence that we are confident about is the stromatolite-based evidence that goes back to about 3.5 to 3.6 billion years ago. Late heavy bombardment, a small bunch of planetary scientists in the field, a very small minority bunch, are actually even questioning if late heavy bombardment happened the way we understand it. But that aside, whatever interesting possibilities were chemically interesting possibilities that was allowing for this transition from inorganic chemistry to organic chemistry to biochemistry, whatever might have been possible prior to this period, where we nevertheless know that there was a lot of these collisions going on. You're right, a lot of it would have been wiped out. What might have still survived is what might have been hidden away in certain environments where probably getting to them would have not been readily possible. But there is a good chance, and I think this is something that we all agree with, that it might have reset a lot of the initial interesting possibilities that might have happened. But it nevertheless brought in a lot of amazing fundamental ingredients that nevertheless were readily available for once again kickstarting the process. So exactly what happened and what got lost and what got reset and restarted again is very, very unclear. Will we ever be able to figure out what exactly happened? The answer is probably no.
Simran is asking which computational tool is used to study the protocellular membranes?
So Simran, I need to ask your forgiveness because I'm not an expert in this at all. However, there is a person that you can look up. Her name is Janet Ivasa. And Janet Ivasa worked with, as a postdoctoral fellow with Jack Shostak, one of the very important leaders in the field, to actually model some of these protocellular entities. She was one of the first people to systematically do it and came up with these gorgeous animations that I think Jack still hosts on his website. So if you kind of go to that site, and Janet runs her own lab now, if I'm not wrong, she's in Oregon. So you can go look up Janet Ivasa, Iwasa. Please look her up, and maybe her site gives you more details about the kind of computational tools she uses to model these entities. Please remember, these are all multi-atomic and molecular-based simulations, so they are computationally intensive. And the tools are varied. Some people come up with their own tools, and some people use pre-existing tools like, I don't know, for example, if BioRender will allow you to model some of this, I'm not sure. You can surely come up with cartoons, but I don't know if you can go from there to actually modeling some of these processes. But please, Janet Ivasa, look her up and you will find more answers, is my feeling.
And if anyone needs spelling for that, we can put it in the Discord as well. Sure. So another question from Neri. She's asking, when it comes to finding diagnostic biosignatures in environments like those of Titans, is finding units functioning like RU, TC, and IL in proto-RNA enough, or do we also need some more information?
What you will primarily need is to have some sense of the fundamental chemical entities that is present in Titan's environment. I'm going to use Titan as an example. One of the things we do know about Titan is it probably hosted water in its past, though we don't really see its signatures right away. But we do see signatures for benzene and a lot of other chemical molecules, carbon-based molecules, and a molecule which was for the first time described in Titan's atmosphere, which modern chemists don't even know about. It is cyclo blah, 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 blah. I don't know the name. I'm so sorry. It's very complicated. I can't remember it, but it's this 3-carbon molecule, which they think would have been a precursor for forming more complex carbon molecular structures. So what is important to know is important to have a sense of whether it's Titan, whether it's Enceladus, whether it is Europa, is some notion of what the molecular space is in that particular system. And that sort of will give us a better sense of what to probably look for. But having said that, if you look at, say, for example, the proto-RNA, we know what the fundamental atoms are that it is made of. So given that the interstellar medium chemistry is the same, and a lot of this bombardment that brought interesting organics onto early Earth would have also been very similar in the molecules that rode on other meteorites and comets that bombarded these other bodies might be similar. There is reasonable justification to assume that it's the same inventory of atomic molecules there. So maybe the fundamental molecules present there might be reasonably similar. But to be able to think about more complex molecules, you need to have some minimal sense of what the fundamental simpler entities, molecular entities might be, what that inventory might look like. It's better to have some sense of it. And happy to say that Clipper that looked at Europa's environment and stuff has given us some sense of what are the molecules that are present out there. Oh no, I'm so sorry. Clipper is a future mission going out to Europa. Cassini, if I'm not wrong, was what gave us, Cassini's results has given us some sense of the molecules that are present in these environments. So we have with some confidence, we can feed in data that look for some of these molecules in a systematic manner. However, the field of biosignatures is still sort of up and coming. We only talk mostly of biosignatures, whereas the work that happens in my lab and in few other labs in the world is insisting on the fact that you need to also look for A biosignatures, which probably will give you a sense of whether that particular planet or moon is actually on the way, is actually in the process of, is actually at a stage in time where life has not yet emerged, but maybe it's just getting ready to allow for this transition to happen. And as of now, the way we look at it, we only look for biomolecules. So we might be missing a lot of other interesting things that might be happening on some of these entities if we don't look for A biosignatures more systematically. Long-winded answer, sorry.
No, it's very comprehensive. Thank you. So let's wrap up with one more question that came in from SM. Do you believe that life universally is made of molecules with both nonpolar and polar parts, as on Earth? Or would it be possible for life to exist in purely nonpolar or hydrocarbon-based environments, such as on Titan?
I will use one example. I would find it very difficult to think about how you can compartmentalize things if you did not have molecules that had both these features. For the sake of argument, let's say, hey, Sudha, you don't need vesicular compartments. You can probably have porous rock surfaces providing environments where you can have interesting chemistry happen. Possible. But having said that, how do you go from that porous rock-like entity where some initial steps did come about to undergoing a process like Darwinian evolution, which acts on sets of populations of molecules that are interacting with each other in a much larger scale? I have a bit of difficulty wrapping my head around that. So I feel like you would have had to have molecules that had several different interesting properties to facilitate many of these important events that lead to the hallmark features of life, at least as we understand it, based on the biochemistry we see on Earth.
Yeah, no, that's great. Yeah, I don't know if you've read the work on azotosomes, but some people are thinking about like the inverted membranes where you would have the hydrocarbon, like the nonpolar head, which would make contact with the nonpolar liquid, and then a polar tail. But it's definitely very like theoretical still.
But even then you have both these entities in that molecule there.
Yeah, exactly. Yeah.
Yeah, it's just not only non-polar, right? So that is the catch.
Yeah.
And that is why I think it's a bit difficult to think about only a hydrocarbon-based environment.
Agreed. Yeah. Okay. Well, thank you very much for sharing your time and expertise with us. That was a really inspiring talk, and I think we all learned a lot.
Thanks a lot. Thanks for having me.
All right. Thank you. All right. Bye, everyone.